Hello, and a big warm welcome to everybody joining us today for today's ISNTD Connect session, Digital Diagnostics Technology and the Transformation of Healthcare Delivery within Africa. I'm joined today by Dr. Francis Krampa from the West African Center for Cell Biology of Infectious Pathogens at the University of Ghana, and Dr. Nicholas Moser, a research associate at the Center of Bio-Inspired Technology, Imperial College London. Before we get into the session itself, I just wanted to give you a bit of context in terms of why we're kind of looking at this area of digital diagnostics. From our point of view, at least, we really felt that the COVID-19 crisis, we're all kind of living this, isn't just affecting our individual lives in terms of the restrictions. I know we're about to come out of lock in the UK, at least, hopefully by the middle of or the end of July. But the worldwide crisis in terms of the economic crisis, some countries are facing are worse than other countries. What has happened is that this crisis has triggered certain shifts in terms of, let's say, employment, modes of employment. We're working from home. Certain sectors of the economy have collapsed. It's kind of shifts, the COVID shifts have taken us to an inflection point, really, where we are able to kind of reimagine a world, as it were, new prospects, where we're able to build in resilience, bake that into the healthcare system. We're able to generate economic growth. The trick is to keep that inclusive for everybody so that we can hopefully impact on the vicious cycle of disease, poverty, disease. And certainly that's a key area that we're kind of looking at. Some of the trends that are driving, from within the trends that are driving this kind of sustainable inclusive growth, one of the major trends is that of digitalization. So the digital revolution, we wanted to kind of have a look at how that's going to impact our kind of space of global health. We struck up a very strong relationship with the Digital Diagnostics Network for Africa group, led by Dr. Aubrey Cunnington, and with Francesca Pfeiffer at Imperial College London. We actually gave them an award, a recognition award, at our Science Communication Festival earlier in the year to do the piece of artwork that they were using to translate the very complex lab on a chip project they were working on into something that could be understandable to a wider kind of audience. From that point onwards, we really felt that this is a major issue that needs to be kind of delved into. So we're very happy that we've worked with Aubrey and Francesca to put together a series, and today is the first in that series. And I'm very honored and delighted to be joined today by Dr. Francis Krampa from the University of Ghana and Dr. Nicholas Moser from the Imperial College London. So I think that's enough from myself. We'll hand the floor over in a second. What I would ask is from the audience, if you've got a chat function there, some of you are already using that. If you could add your name, just say a quick hello and country and organization, that would be great. We'll give you a shout out. And that's Dr. Stephen Bramia from Canada has joined us, and he's saying hi to everybody. So hello there, Stephen. Nice to see you there. Feel free, guys, to put in there. Francesca Pfeiffer, hi everybody, delighted to be here. It'd be a good kind of way of communicating through this, as well as putting your own questions in, which we will weave into the Q&A after. So I think that's, without further ado, that's enough from me. And I'd like to pass the floor over to Dr. Francis Krampa from the West African Center for Cell Biology of Infectious Pathogens, University of Ghana. Francis, over to you.
Yeah, sure. So thank you very much, Cameron. I'm really happy to have been invited to share my thoughts with you. A very good afternoon to all who have tuned in, and I'm particularly grateful to the ISNTD and the Digital Diagnostics for Africa Network, who give me the opportunity to represent them in giving this talk. So my name is Francis Krampa, and I am a fellow at the West African Center for Cell Biology and Infectious Pathogens. So we are going to discuss briefly the continuing need for point-of-care diagnostics in Africa, I hope to be sharing some insights and my thoughts with you. So the African population, particularly sub-Saharan Africa, is made up of about 1.1 billion people. And this is a continent who is no stranger to infectious diseases. You have lots of infectious diseases, non-communicable diseases, etc. in here. And you find out that majority of the population live in rural areas. About close to 65-70% of the population live in rural areas. However, the unfortunate news is that all the sophisticated and the well-equipped healthcare facilities are clustered in rural areas. So this leaves a huge deficit and a huge gap in healthcare, especially when it comes to diagnostics. To give a clearer picture, if you find yourself in an urban area, you are within 1 to 12 hours away from primary healthcare, whereas just in about 1 day or 2 days, you should get access to all sorts of referral laboratory confirmatory tests that you would require. On the contrary, if you live in a rural area, you are close to 1 to 4 days away from primary healthcare and another 5 days away from a referral laboratory. Now, the bad news for some people who live in extremely remote areas is that the only time they have access to seeking laboratory healthcare is when corporate bodies like churches or like government agencies visit their communities to do basic screening of certain endemic diseases. So the challenges in laboratory diagnosis in Africa are diverse, but I've clustered these into 3, which would be accessibility, quality, and implementation methods. Now, since we have a diverse group listening, I would want to say that, in fact, there are so many commercially available diagnostic kits, but nearly all of these are not accessible in most parts of Africa. Now, when it comes to accessibility, there are troubling issues such as lack of trained personnel. You don't have enough laboratory personnel who are trained enough to operate diagnostic equipment. In fact, reading a report from, I think, the WHO a few days ago, they made mention that lots of test devices, kits were shipped to Africa through donor agencies, but they didn't have enough qualified people to operate these. Therefore, they were relying on gathering dust. There are also no laboratory facility infrastructure. People who seek healthcare have to travel long distances to nearest laboratory. And in these regions, people tend to pay for the health themselves. There are no insurances that covers in the National Health Insurance Scheme. So the high cost to patients deters them from seeking healthcare and then laboratory diagnosis. There's also lack of poor health-seeking behavior. And when this happens is that because people don't seek behavior, there is a kind of non-trust to invest in these, because people who invest in these tend to be afraid that they are not going to get returns, so they tend not to invest in private laboratories or Next will be quality. You tend to see that labs are not very equipped to run very robust methods. The labs are not properly set up. There is absence of generally good laboratory practices. They don't have poor maintenance culture. And then some products come with very unclear guidelines that they tend not to be able to be used properly. The last will be policy, which is merely implementation. There tends to be poor implementations of diagnostics into the general healthcare system. The supply chain issues, cold chain, things that have to be kept cold, cannot be kept cold because of electricity, power, and infrastructure. So these further compound the healthcare. So the implications of these is that you tend to have this divide in labs. You tend to have very under-resourced labs, which have no laboratory facilities, very minimal. And you have lots of moderate advanced labs, but they are all clustered in urban areas. Now, in terms of infrastructure, those in the moderate to advanced labs, they have dependable electricity, they have cold storage, clean water, trained personnel, etc., dedicated lab space. Whereas if you look at the under-resourced laboratories, they basically have no electricity, no cold storage, lack of clean water, and no sterile environment. Look at the kind of samples these two labs can analyze. The people who lived in under-resourced areas can only do finger prick at best and nothing. It's impossible to interrogate CSF. It's even impossible to get sputum for culture or anything. Whereas if you come to the moderate to advanced labs, they have all the technology to do anything from complex samples to very easy to access samples. They can do everything. Now, so the under-resourced labs are left with no choice but to run very basic tests like just simple lateral flow assays, and that's it. They can do optical imaging techniques, they can do molecular techniques, and they can't run serological techniques. Whereas the moderate to advanced labs have the capacity to run all these things, but only for the select few who live in urban areas or can afford these tests. However, notwithstanding, we cannot deny the importance of diagnostics. Diagnostics is very necessary for appropriate therapy, for real-time data in case we want to stratify the efficiency and effective disease control we need to diagnose, be able to detect and set control measures. Because of this, surveillance is usually compromised. We don't tend to, have good surveillance systems. And in this era where drug resistance has become a very, very pressing issue, we tend to do lots of presumptive treatment because, most facilities cannot properly diagnose, therefore they just, have to do presumptive treatment. And due to this, there could also be a rise in drug emergence, and we tend not to be prepared for outbreaks. Now, what would it take to accelerate the fight against infectious diseases? Now, I've put these in 6 categories. The first would be to develop sample low-cost tests for use by first contact healthcare providers, and these usually need to be as rollout tests. We also need tests that can do multiplexed and then multi-analyte testing, so patients wouldn't have to run this test and that test and then another test. There's also the need to have rapid drug resistance susceptibility tests. We should also learn to get new biomarkers in to assist us replace tests for specimens which are difficult to obtain. Now, there are also very highly sensitive molecular tests that span through the value chain. However, although these are quite expensive to run, in the long term, we could learn to develop cheaper, tests for these molecular tests. Now, the most important is that we need to do a close to patient diagnosis, a diagnosis which is closer to the patient. And that is what we go to next, which is point of care testing. So the barriers we need to breach in obtaining this crucial next generation diagnostics is that developers should learn to create tests which are not reliant on electricity alone. We need to get equipment free. All tests have to have solar rechargeable or battery operated devices. The cold chain storage also has to learn to be broken since there is no electricity. There tend to be lack of freezers for reagent storage or -80 freezers to store some specimen for later analysis. So if possible, developers need to integrate the test processes into fast, simple lab-on-chip devices. Because there's no clean water as well, this lab-on-chip or cartridge-based devices are very promising to develop as point-of-care tests for Africa. To address lack of trained personnel, we need to go towards automation in this era of digitization. We need to look more at automation to make tests which are very, very automated. You don't need, the technician to be properly trained in every assay step or have to do multi-steps before they get to the result. We need to move into automation to get minimally trained people to operate these tests. Also, sample to volume analysis, because of childhood illnesses and then patients not being able to provide lots of samples, we need to probe analytes in samples which are very easy to obtain, and if possible, non-invasive testing. Turnaround time recommended test should be looking around having the test done in an hour. Footprint and costs, the devices shouldn't be really expensive for easy uptake and adoption in low to middle income countries. In terms of data storage, the digital platforms of today would allow us to do lots of data transfer to track and keep an eye on the trends of data movement, where data is going. So this brings me to a very critical point in this. So we're talking of diagnostics, and we are narrowing into point-of-care diagnostics. So to better understand it, I've put in very huge trivia. If in case we'd like to tell time, how would we want time told? Well, we could use a sun scale or the hourglass or a clock done something. But what if we need to tell the time in real time? Then we either have a pocket watch, a watch, or even a Fitbit. So the difference between these is that they all tell the time, but these ones are close to us, and we can tell the time. Even if we are sleeping, you can just pull by your bedside, take your watch and have it seen. However, if you decide to go by the old-fashioned way, you'd have to wait, probably go downstairs to be able to tell the time and come back to bed. In case you need to take your medications at the time, you can keep an eye on real time. You need to always go look for the time and come. So point of care testing is like telling the time in real time, having the watch close to you to be able to always tell the time. Now, in 2003, the WHO came with guidelines that we need to adopt in developing the point of care, the next generation of point of care diagnostics, which was the Assured. Yeah. However, since then, there seems to be a skewness towards HIV and malaria diagnostics. These have really transformed HIV and then malaria diagnostics. But it seems that lots of the other diseases have not received equal attention. For instance, NTDs, point of care, are still lacking in nearly all STDs, which keeps being a burden in sub-Saharan Africa. For even more prevalent diseases, infections, such as STIs, you only find that the predominant point of care testing is syphilis. Others like gonorrhea, chlamydia, HSV, Haemophilus ducreyi, etc., tend to be lacking. But let's not forget that these are all risk factors for HIV. So if we can't diagnose these, we'll silently keep transmitting the HIV without really knowing. Fevers, for instance, we hardly have lots of tests that can detect bacterial biomarkers, or even differentiate one fever from the other. So many fevers present with the same symptoms, and because probably malaria is more endemic in most sub-Saharan African regions, you tend to have doctors overly prescribing anti-malarial drugs because they probably just suspect it's malaria, but these drugs are often being abused and without tackling the exact roots or cause of the disease. Therefore, the next generation point of care are trying to look at using microfluidics to interrogate lots of markers, host biomarkers, etc. And with microfluidics, with a small sample, you could interrogate so many analytes in your sample. We're also trying to look at how nucleic acid-based testing could be integrated into point-of-care testing. And combining these with digital diagnostics gives a really powerful tool that we can use in facing future of diagnostics and making robust, good diagnostics for point-of-care testing in sub-Saharan African regions who do not have the capacity to either routinely run PCR or do any advanced molecular biology testing. So how could this digitization be included and then coupled with nucleic acid testing? So we all know how the glucometer functions. So let's imagine a device like the glucometer, for instance, or these popular Fitbits that we wear. Supposing we had some of these to routinely diagnose any infectious disease. I mean, even my grandmother, who is close to 80, can test her own blood sugar, look at where the label, maybe red, green, is, just tell me that, oh, the small device is telling her that her blood sugar is high or her blood sugar is low. And she does this with ease without, any help. What if we had certain glucometer-like tools that could, just use these simple platforms to interrogate biomarkers, which can detect diseases prevalent in that area? That would really be great and good for point of care testing in Africa. Now, let's not forget that the clinical cause of infection is you get exposed to the pathogen replicates in you before you start an immune response. However, because most of the point of care testing, which are based on lateral flow immunoassays, they detect antigens and antibodies in sub-Saharan Africa. Many of them are based on human immune response detection. You tend to get this often late, by which time spread has continued. So what we need to focus more is either trying to get a pathogen, which will take very heavy laboratory work. It means the patient has to go to the hospital to do either microscopy or nucleic acid testing. However, these cannot be done without going to laboratory. But what if we could have devices that could actually start interrogating either some biomarkers or some nucleic acid traces in the early stages? And mind you, with several years of exposure, there are now lots of carriers of infectious diseases who do not show symptoms. So symptomatically, they are all right, but they carry the pathogens and they keep replicating these and then transmitting, which is not good for public health. So we could take a huge lesson from the clinic, from the evolution of molecular diagnostics. When I say molecular diagnostics, forgive me, but I mean nucleic acid testing. Now, back in the 80s when it started, there were lots of heavy equipment. Equipment were, really big. And today we have sample to answer platforms. Although these are not widely, distributed in Africa, but you have so many point of care tests, not in Africa anyway, but several prototypes nowadays that can do small gene sequence detections and stuff, all using just handheld devices. So this is a real model that, going forward, point of care testing should follow and get self-contained devices that can do robust and very accurate diagnostics, as well as being able to multiplex them. Some of its advantages include that they can even detect in low transmission areas. They are very good for detection of infections in low transmission areas. They can be multiplexed. They could even detect mutations. So sometimes mutations can cause antigens to be modified, and lateral flow assays cannot detect them. These platforms would enable detection at the gene level, detecting exactly what the infection is and exactly the mutation, arising from that. That's why it's not being detected. When we do these, it would help us avoid overtreatment and mistreatment, which often leads to, drug resistance. It will also reduce the burden of having to do an extra diagnostics. So a patient wouldn't have to run 3, 4, or 5 tests. With just one device, he runs a multiple array of tests, and he is okay to go home. So it might look expensive, but in the end, it's cheaper because you can run more with less. The potential of coupling digital diagnostics with these evolution in nucleic acid testing is that I've already talked about the self-contained devices and the multiplexing capabilities. One good thing is that they rely for real-time analysis. you do it there, you get your test, you get your results there. Next, they could even tell you, okay, probably because we are seeing genetic material, it means you still have the active, pathogen in you. It is not like, okay, it's passive, you've cleared the infection, but you still have the viral antigens or the viral antibodies in you, or you keep responding to the antigens, that's why you have antibodies in you. They could also allow for qualitative results, which generally protein or antibody-based detection systems do not let us do, plus data storage and connectivity to seek remote help, sorry. One other very important thing is that combining digital diagnostics with nucleic acid testing is that, well, forgive the little cartoon, but there seems to be a long-standing feud between nucleic acid tests and then immunoassays, each claiming superiority over the other. But diagnostics is different. We could detect active disease, we could detect exposure, we could also detect immunity or even specific traits. Each party is strong or it's good at doing one. So these platforms could also help us, combine both. So together they can do they can perform a diagnostic and tell you exactly whether it's just an active case, or you have been exposed, or you have built immunity, or even detect a specific strain. So a combination of several types of methods is possible through digital diagnostics. Now, the good news for Africa is that we wouldn't need to reinvent the wheel. In fact, reports from the African Union and the World Bank states that Africa is growing at a very fast rate, aided by digital transformation, which is accelerating growth enormously. And the technology is already there. There are several prototypes that we don't have to reinvent, just try and get new biomarkers, and then repurpose those platforms to suit the requirements of diagnostics in Africa. Yeah. So in conclusion, I would like to say that point-of-care testing cannot replace laboratory testing. However, if we make point-of-care testing available, it would probably, or even more, give access to about 45% of the African population, which has not already been covered by diagnostics. Yeah. So we either need to embrace technology, or we fall behind completely. There's a huge chance for us to leapfrog using technology. Now, and we can do this through the collaborative partnerships, through systems that have already been set up, and then scientists who have already built all these for us. Yeah. So I would like to acknowledge the network to which I belong, the Digital Diagnostics for Africa Network, who made it possible for me to give this talk today. Thank you very much.
Thank you very, very much, Francis, for a wonderful presentation there. A lot to unpack and a lot to think. I especially like the closing slides in terms of the economic potential for digital diagnostics, underlined by the African Development Bank themselves. And I know in the audience we have Dr. Maimouna Diop. Hello there, Dr. Diop, from the African Development Bank, but based in Abidjan, the Côte d'Ivoire, amongst the audience. So I myself was going to pick up on something the AFD, the African Development Bank, put out in May and June in 21, looking at entrepreneurship, alliances for entrepreneurs within Africa, some of the recommendations of that report. We'll probably weave those into the Q&A at the end. But thank you very, very much, Francis, for a wonderful presentation there. And I would really encourage the audience to throw in some of your questions in terms of the chat box. We're having some interesting questions coming through, which we'll come to towards after the next presentation. But thank you for that, Francis, a lot. That brings us to the next speaker, Dr. Nicholas Moser, who's very kindly joining us today from sunny London here. He's a Centre by Inspired Technology at Imperial College London. So I think over to you, Nicholas, and if I could ask Francis and myself, we'll just back out in terms of the things. Over to you. Thanks a lot.
Perfect. Thank you very much. Can you hear me well? Just check.
Yeah, perfect. We can hear you and see you perfectly. Thanks. Yeah.
Perfect. That's great. Well, thank you, Francis, for this great talk. It's definitely been really important to set up the actual demand in Africa for new diagnostic technology. And perhaps a very quick introduction. So I'm Dr. Nicholas Moser. I'm actually originally an electronic engineer, and I'm also, just like Francis, part of the Digital Diagnostic for Africa network, which tells you a bit about the multidisciplinary background of all the members of the network, which is so important when trying to develop new technology to make an impact in diagnostics. So I'll tell you a bit about these potential new technologies for molecular diagnostics. I've included a title, Point of Need, whereas Francis talked a lot about point of care. And that's for the idea of trying to go the extra mile, trying to bring it directly, to villages, to remote settings, because that's the reality at the moment. We're at the brink of we are very well within a pandemic at the moment. We've got diagnostic issues here in the UK, but in Africa, there's nothing new. There's been a huge need for diagnostics at the point of care, at the point of need, in low resource settings for pathogens such as malaria, dengue, and particularly in Africa, what we're looking at with the network is malaria. So at the moment, as Francis described very well, and I want to go through these very quickly, there's only a few possibilities for actually carrying out a diagnosis anywhere. And the very first one is, of course, cultures, which are often what labs are limited to. And that's something that came out of talks within the network, within also NGOs, that often, cultures are what's available. And they're, of course, very sensitive. They are, however, they do take a lot of time. They require expertise and they require a lab. And then there's the other end of the spectrum, which is, of course, the rapid tests. Very rapid. They require no expertise. They're highly portable, highly affordable. However, they're not qualitative. And the antibody ones are both infections, and the antigen tests, they usually have very variable sensitivity. And then there's what I would call the gold standards for detection, which is the molecular methods, typically your RT-PCR method, which has time of several hours, requires expertise. It still does require lab, high cost, but it's very high sensitivity, high specificity, and highly quantitative. So the question that I'd like to touch upon in this talk is, can we get the best of both worlds? And it's a problem that we're having now in the UK as well. How can we diagnose diseases highly accurately, but also portably? And the answer so far has been, here at least, the antigen test. That's what's being used for mass testing if we look at the COVID pandemic. But as you're very well aware, looking at how the pathogen presence evolves in time, the antigen tests have a much higher limit of detection than the molecular tests actually do. And that's what we're trying to get to. We're trying to detect the disease as early as possible as it arrives, which, of course, molecular detection is more helpful with. So as I said, what we really need is trying to combine the accuracy of the molecular tests with the portability and scalability of antigen tests. And something that also Francis has mentioned is the World Health Organization helped us define what's needed for a diagnostic test. And as engineers, we really like that. We like to be given specification. And this actually started with sexually transmitted diseases. And it's a short criteria, just to go slightly over it, affordable, sensitive, specific, user-friendly, rapid, robust, equipment-free, and deliverable to end users. And since then, we've added the idea of the digital connectivity as well for surveillance, which is so important when, deploying and mass testing in continents such as Asia or Africa. So looking at all these requirements, we need to come up with a technology that is highly scalable, but also low cost. And here are some other fields where we've actually managed to do that. If you look at how technology has advanced over the past decades, you'll have a mobile phone, you'll have computers, and they've become several orders of magnitude smaller over the past years. And this has been this has been enabled by the emergence of microchips and the evolution within the years. So here's a question. Can we apply microchips, which has had trillions of dollars of investment over the past decade, to issues that we have nowadays in healthcare, and particularly diagnostics? And I want to look a little bit into what happened in the field of microchips, what allowed you to get, such small computers and smartphones. And, the building blocks of electronic is called the transistor. It's a device. And originally, in the 20th century, it was a very large device. And every year it actually gets reduced in sizes. And now there are over 100 billion transistors in your mobile phone. So this is how many devices you can put in one. The technology has gone down to feature sizes of 7 nanometers, and they're able to perform trillions of operations per second. That's a huge computing power in such a small scale. And in fact, that's been enabled by a law called Moore's law, which says that the number of transistors in a dense integrated circuit doubles about every 2 years. In short, since the 70s, we've been able to put more and more capabilities within the same small microchip. Well, that's great, because in a way, what we're trying to do now is develop new systems that are highly scalable and highly low cost, which you can see on this graph is what microchips are enabling. So how can we transfer the technology that is microchip to the problem of diagnostics? And that's kind of been part of the question that we're working on solving in Dr. Georgiou's lab. So the lab really builds upon 3 pillars to design new bio-inspired devices in the form of integrated sensing systems. So having on those microchip sensors, as I'm going to show you in just a moment, and perhaps integrate them as part of wearable devices to, for instance, track and monitor symptoms. And at the core of it all is really lab-on-chip technology, which is very much at the interface of 2 disciplines. On the bottom side of this slide, you've got electronics, microchips, instrumentation, again, what's in your phone. But on the top of it, you've got molecular biology, where you aim and target biomarkers that you want to detect. And that's very broad, of course. Let me give you an example of what biomarker we could be looking at. So here on the left is a microchip. That's the one I showed you. And it has these sensors here, which you can actually kind of see as a camera. So, your camera has thousands of pixels. They're like sensors that capture light. But what these sensors are, so these little square, they're also pixels, but instead of capturing light, they capture ionic activity. So you put a solution on top of it, create a reaction, and you're actually imaging this reaction as it happens. One example, and this particular example is actually ionic solutions in terms of potassium. We create a change of potassium only in parts of the array, and you can see, just as you would see on a camera, you can see the ionic activity at the surface. So it really transfers chemical reaction into an electrical signal which can be processed. And that's what lab-on-chip is all about. And that biomarker, as you can imagine now, because we're looking at molecular testing, we're targeting DNA and RNA for detection at the surface of the chip. And the way we're doing that is we're causing a reaction of DNA amplification similar to PCR, but as the reaction happens, it will release protons, H+ ions. There's a pH variation. And on the electronics, we implement thousands of sensors on the microchip, which are able to pick up this change in pH and provide a signal which will tell the system whether the DNA is present, and not just that, also the quantitative detection of the RNA or the DNA. This can then be connected onto a platform that might be portable, but also sync to a cloud server for data. Let me go a little bit more into all the steps of this approach. So microchip is ideally suited to particularly DNA diagnostics at the point of care, because as I've said, it's in your phone. It can produce small handheld device without the need for bulky optics. There's no need for what your large instruments in the lab would use, which is an optical source or optical detectors. In this case, the detection is actually electrochemical in a very small volume. The detection can be done in real time with integrated instrumentation, and over 1,000 sensors are actually integrated within the small microchip. And we've improved in the group that it actually allows for reliable DNA detection. A bit about the chemistry that we're using. If you're familiar with PCR, the approach is actually similar, but the molecular chemistry is called LAMP, isothermal, loop-mediated isothermal amplification. It does not require expensive instruments because it's actually an isothermal method, as the name indicates. There's no need for thermal cycling. Highly specific because it requires 6 to 8 primers, which is more than what PCR does. And it's also extremely fast with a positive detection in under 10 minutes. And as I've said before, during nucleotide incorporation, protons get released and can be picked up by the sensor, which is the way that we can detect the signal associated with DNA amplification. This is a bit of an insight on the chip that we've made. So the chip is called Titanics because you know this by now. My name's Nick, and we're actually 2 PhD students back in the day who designed it, both called Nicks. So yeah, we decided to call it Titanics, which is the name of a sinking ship, but I like to say it went down in history. Whether it did or not, I'll let you judge. So the chip has several thousands of sensors on it, and it's that rectangle that you can see here. And in terms of scale, it's important to realize that the dimension of the chip is 4 by 4 millimeter. And you can see on my camera, this is what it looks like. So that's the chip here in the middle. It won't really focus on it, but you know, it's a tiny dimension. And of course, it's quite counterintuitive to imagine that you're going to start putting liquids on the surface of electronics, because you very well know that they don't go together well. So you need microfluidic approach, you need some sort of encapsulation method to be able to pair both. And in the group, we've started working on 3D printing prototyping for such approach. And we've come up with a design of manifold to place on the surface of the chip with 2 wells, one for a control reaction, one for a sample. The control, for instance, could be a negative control to test against amplification, or the positive control to test that the sample's properly been taken. This means that the overall volume of every chamber is around 5 microliters. And once again, I'm showing on the camera the manifold that we've designed on 3D printing. And it's the same kind of dimensions I've shown before. So we've come up with a whole workflow solution, and this particular graph shows for swaps, but of course we're working on several sample types. I don't want to go into the sample processing module in this particular talk, but we can discuss in the questions. What I guess I want to focus on is really the detection principle, where we're detecting the pathogens in this case 2 wells, sending the signal directly onto your smartphone through an app via Bluetooth, and then provide connectivity to a cloud. And this is actually what the handheld device looks like. So you've already seen the cartridge, which is where the microchip is present. It is actually nothing else than the microchip and the manifold, which makes this extremely low cost and highly scalable, as I said at the start of the talk, because all these components are actually very standard. And we've paired it with a handheld device, which we've called Lacewing. And I'm also showing this on my screen to be compared with my phone, you know, very similar format. And it's a handheld device where the cartridge is plugged here in the middle. And what you can see here underneath is a heater, which will allow to perform temperature regulation at 63 degrees for the LAMP assay to happen at the surface of the chip. And under 30 minutes, the time is of course variable on the assay, we can detect the presence of the DNA in real time, but also quantitatively, and send to a cloud server such that it can also be geotagged for surveillance. You know, I'll show on the later slide the interface for the dashboard as well. Perhaps just a quick image to show you how we use this in the lab. This is very much in the lab, but you can see the unit here in the middle, and you can see how we pair it onto a very standard smartphone application. We plug the cartridge here in the middle, which makes all the electrical connection with the microchip. And then perhaps I want to pause on this particular site. So here what you see on the screen is the image of all the sensors, and you can discern 2 shapes here, which are actually the 2 wells. So we'll be running a positive reaction here, a negative reaction here, and we'll be able to extract the information from these 2 reactions, similarly to your qPCR instrument. So we'd load the reaction, and then in real time, the data from all the sensor is being streamed directly on the smartphone app, which you're going to be seeing here. And we can extract the signal and provide positive or negative results. So we've shown in the past that the results are very much similar to your standard qPCR instruments. From the data, we can extract the natural amplification curve, which will give you information also on the viral load of the sample. But of course, looking at doing that in a handheld fashion at the point of care rather than in instruments. We've proven this for several pathogens and throughout the world. So actually the first trial that we did was in 2018 in Taiwan, which is the first time we took the device out of the lab. Then last year with partners at the network, we actually tested this in Ghana for malaria. But in the UK, we're also working on antimicrobial resistance, aspergillus testing. In South Africa, we're working on tuberculosis. So there's quite a lot of projects that we're involved in. In a way, perhaps this is very counterintuitive to biologists, but as an electronic engineer, the focus is really on the detection method. After that, it's down to the assay to make it specific to the particular pathogen. Just another, perhaps a quick video to show you some of our PhD students in the lab who actually are at the talk today. So thank you, Vanna and Kenny, using the device, several devices actually in Ghana in the labs, which is where we tested it with clinical samples for malaria and proved detection in under 15 minutes. All right, but perhaps just a few notes. Of course, everything changed for us, just like it did for many people, March last year when COVID came around, and we kind of saw this as a good opportunity to work on technology and demonstrate it for COVID, for SARS-CoV-2 detection. So that's where we actually did a lot of progress on the tech, but also demonstrated a detection of SARS-CoV-2 in under 20 minutes. And just a few graphs showing you, this is a RT-QLAMP assay versus a standard CDC RT-qPCR assay with good correlation for quantification. And this is the correlation of the time to positive between the instruments, so the RT-qPCR instrument in the lab, and lacewing or handheld device, showing once again good correlation between the results. And we actually demonstrated sensitivity of 97%, specificity 100%, and average time to results of under 13 minutes for the samples that we did here. We tested 34 samples on the handheld device and over 100 samples in the RT-PCR machine. Very quickly, some of the signal processing happening behind. So, the sensor output is actually processed such that the DNA or RNA signal can be extracted to provide the positive result and a negative result, and all this on the same cartridge. So this comes from the sample well, this comes from the control well. So every test can come up with, whether it's valid or not based on whether the control is behaving as we expect. So overall, I guess the aim has been to put diagnostic capabilities in the palm of your hands with technology detecting under 20 minutes. But there's also whole work to be done in the back end, of course. So we've developed a very early version of a dashboard as well to allow to visualize all the data that's being sent onto the cloud server. And these are, of course, just mock-ups, but it's important that all the data can be pulled through and provided to decision makers in real time, rather than what's being done for COVID, where it's a lot of manual entering. So we're actually in the middle of a DHSC validation at the moment, testing with 410 clinical samples, and we've tested at the front end at the moment, gotten really encouraging results. But I hope that in the next few months, we can prove, we can finish this validation, provide some really good results to everyone here, and then go in, ask the question, how can this be applied further to low-income countries, and use these great results towards malaria detection as well. So overall, the goal of point of care has been to bring electronics and diagnostics together in the palm of your hand with, as I said, the versatility of being able to swap cartridges for different pathogens as well. There are some new avenues of research. For instance, I've just made a new chip, which I've called Skywalker, which has now a lot more area and more pixels, with the advantage that we can have more than just 2 wells and be testing a panel of infections on the same cartridge, potentially 4 serotypes of dengue, potentially malaria, and antimicrobial resistance genes. So that's something that we're looking at the moment, how to implement this within the same test. And there's quite a lot of research going on into kind of sensor learning and AI algorithms to try to extract the signal faster, such that ultimately we'd be able to, not detect the infection at the peak here, but as early as possible, so that we can even more and more prevent pandemics or outbreaks and attack it early to allow real case identification. So I'd like to acknowledge Dr. George's group at the Center for Bioscience Technology. As I said, several are here now, so thank you very much for that. And of course, the Digital Diagnostic for Africa network, of which I've been part for over a year now. It's been really good to, as an electronic engineer, be involved in a network of clinicians and find out what the need really is. Thank you very much.
Thank you very, very much, Nicholas, for that amazing presentation. Some great feedback from the audience coming through as well. Great to see the setup there, and I love the way you compared that to your phone as well, showing how easy it is to use and everything. So appreciate that a lot. Thank you. If I could ask Francis and Nicholas just to both come back, we can start the Q&A. Some really good points coming through from the audience. And I'm trying to weave those into this kind of questions that we're going to be asking as well. I think there are a couple of technical questions that have come through, and I'd like to address those first, out of respect for the people that have asked them. So Dr. Deirdre Joy from the National Institute of Health in the United States is asking, I think this was aimed more towards you, Francis, but I can open that up to Nicholas as well after. She said, excellent talk, thank you. I'm curious as to your thoughts about the adaptation of CRISPR-based diagnostics for infectious disease point-of-care testing in LRS. In theory, at least, CRISPR-based diagnostics could combine simplicity and low cost with the high sensitivity of NATs. What are your views on that, Francis?
Yeah, so I think, let me use my personal experience with LAMP to address this. I think one of the major problems LAMP faced when they were doing a trial that I remember was they were having lots of contaminations, lots and lots of contaminations. Yes. So besides issues with contamination, it would work well if only it is isothermal. Yes. And other than that, lots of the other obstacles will be more political than scientific. Mm-hmm. Yeah.
I think they can actually work. They can actually be complementary.
Yeah.
So probably, I mean, I know it's something we've looked at to also be working on CRISPR and try to, increase performance. So I don't think it's this, rivalry there.
Yeah.
So thank you for that answer, both of you, and I hope that answers your question, Dr. Joy from the NIH. We've had another technical question, which I'll put these together at the beginning, from Derek Robinson at the University of Bordeaux in France. Some pathogens don't leave much DNA in serum or blood, saliva, etc. How easy is it to adapt Titanics, your Titanic system, from LAMP to looking at serum antibody antigen-based assays?
So I guess the first thing I want to say is we're trying to, the detection itself is flexible to sample types in the sense that it's all about the front end, right? So we're working on front end for nasopharyngeal swabs for COVID, working on blood front ends. And of course, as you said, it does depend on the viral load, which will impact sensitivity. The second thing I want to say is we're also looking at an approach for diagnosing using the host response. So if you're aware, not just searching for the pathogen, but rather looking at the way your body's responding to that infection, which will, I believe, help for the sensitivity of the detection. And I think you'll have a talk from Ivana, who's my colleague here, who can go more into that approach. Then getting into antibody and antigen, we're absolutely looking at that. It's actually very early. We're actually at the moment looking for funding to pursue that. But personally, I'm very keen on looking at detecting antigen and antibodies using the same technology, because there is a real opportunity to also be looking at other biomarkers than just DNA and RNA.
Yeah, sorry, Nick. When you say trying to look at the antigens and antibodies, are you trying to do a combo, like combining both, or just one at a time? Combining just antibodies, or you can do both antibodies and DNA, or?
I think what I'm saying is I'm trying to actually reach the implementation of a test for antigen or antibody.
Okay.
Once that's done, the sky's the limit, right? Because then we've got both.
Both, yeah, sure.
So thanks for that answer there. Some lofty ambitions there in terms of the rollout. Very, very good. Lisette van Lieshout from the University of Leiden is asking, very exciting new developments. Do you think that sample preparation is a major limitation? I'm thinking about challenges in the detection of parasite eggs from stool samples. That's from Dr. Lisette van Lieshout in the Netherlands. And I know they're working on a CAA, a specific schisto diagnostic there with a few collaborators, and she's asking really, do you think the sample preparation is a major limitation? She's thinking about challenges in the detection of parasite eggs from stool samples. So over to you, Francis, or both of you, Francis and Nick, both of you to. She's qualified that DNA from eggs. Sorry, she's saying, yeah.
Okay. Well, I think when it comes to like worms and then such parasites, I think the gold standard is always to do microscopy, looking at eggs and stuff. But we could also see that because neglected tropical diseases, there's no much attention now. So the skill in microscopy is actually going down. So you tend to have lots of, microscopists who probably may not be experienced enough to detect all these eggs. So I think it's possible and it's worthwhile integrating, genomic detection and trying to look for, eggs, DNA, and some protein markers from eggs and that. So I think it's possible.
Nicholas, thank you for that, Francis. Nicholas, would you like to add anything to that?
No, I think I mostly agree. I myself haven't worked on other sample types in blood, but definitely sample prep is such a limiting factor, and it's being worked on at the moment. So yeah, definitely important.
That's very interesting. And on this DNA extraction approach, Elias Cabas Pinango, who's working as a PhD student actually in the Poppy Lamberton's group at the University of Glasgow, again in schisto settings, he's asking, thank you both for your presentations. I have a question in his mind, probably for Nicholas, but I'll ask both of you. Is DNA RNA extraction included in the point of care approach, or do we need to extract it conventionally before the chip detection? Again, it's the sample preparation type of question. Yeah,
absolutely. So what the pandemic's allowed us to do actually is finally got to that question of how we can extract the DNA RNA, and we've come up with a way to extract in under 5 minutes with a very frugal, no electricity required methods. And I'm sure in another talk we can get my colleagues to come in and talk about it as well. We're also in parallel investigating ways to extract from blood, but these are still under development and not validated. So what we're doing right now is we're looking at a two-step approach where I've presented step 2, which is the detection. Step 1 will have to be sample processing, which depends on the sample type. So you can see it as, yes, it's not integrated, or you can see it as there's a lot of versatility on what sample type you're going to be using. So there'll be a lot of progress in the next few months onto that side.
Fantastic.
I hope that helps.
Thank you for that. Francis, would you like to add anything in that?
Most of the new upcoming technologies are really advocating for sample to answer. Mm-hmm. So you have a cartridge which has lots of compartments with its own reagents. So you just prepare the sample, they just pipe it and put it there. So you have lots of self-contained cartridges for that purpose. So yes, many technologies are looking at that.
Brilliant.
Doing sample to answer.
And thank you for that, Francis. I wasn't expecting a slew of technical questions, but that's what's happened. I'm going to ask you these. So Dr. Ronald Blanton from Tulane is asking, do LAMP patents, so patents taken out on LAMP, and I love this kind of question, present an obstacle to product development?
It's a very right question. Yeah. I mean, definitely. Actually not, because LAMP patents have expired. There's still remaining patents that I think are expiring this year. So in a way, the timing's been quite bad for LAMP inventors and the COVID pandemic, because now you're seeing all these new devices using LAMP and the patents are expiring.
Okay, and Francis?
Yeah, I don't think I have any additions to that.
Okay, brilliant. So, I mean, that's a very interesting, and that's all part of this reimagining, I think, personally, this reimagining the future and what digital can actually do to that. When we talk to, we have an annual drug discovery, development and diagnostics 2-day event that happens every year, D3, I think we're into our 7th or 8th year of that now. And one of the key things when it comes to diagnostics for NTDs, we always hear 3 obstacles from the diagnostic world: lack of regulatory harmonization across the different countries, the manufacturing base issues, adding to costs and accessibility issues around that, and then the big F word, funding. Right? Now, we've had some questions on funding, and I'm just trying to weave those together. Earlier on, we had a question from Dr. Enoch Boateng. He's at the Czech Institute, Ghanaian, he says, at the Institute of Czech Life Sciences in Czechoslovakia, studying there. And Dr. Boateng was asking, I'm just trying to pull that up. He's making a point about, and this is to do with Francis, your presentation, but obviously I'm going to ask both of you. You indicated a lack of investment in the areas of healthcare.
Yeah.
And according to his knowledge, the molecular diagnostics are comparatively higher in cost than the current gold standard diagnostics that are used. The proposal for molecular diagnostics at the research level is viable. Obviously, it is, as you both demonstrated.
Yeah.
But how do you get stakeholders to invest in the mass production in various health facilities? How do you engage with that? And that's an interesting question, because
yes.
African Development Bank's own report that came out in June 2021, it was a white paper looking at entrepreneurship and free trade, Africa's catalyst for a new era of economic prosperity. They're trying to put the entrepreneurial mindset, the entrepreneur, at the heart of efforts to transform Africa's economic prospects. And in their definition of all this, and to build on this question from Enoch, they've kind of put it into the demand side, i.e. they put a supply side of investment into entrepreneurial activity. And on the demand side of that entrepreneurial activity, they're saying that we have to scale back the expectations of financing and revenue of those particular stakeholders, because what they're saying is innovation and the digital kind of thing alone will not kind of lead to wealth and stability across Africa, but what they're rather concentrating on is digital connectivity, infrastructure, improved human capital, education, access to services. And I suppose the Enoch's questions coming in on the back of all of that, really. How do you engage those stakeholders, those investors, to put money into this and make this happen? What would be your kind of recommendations? I'll ask Francis first.
Okay, well, I would probably say part of the problems of Africa is that we tend to lack because we fail to understand the value of diagnostics. That's why there seems to be that burden. Yeah. So first of all, failure to understand this. Now, usually with investors, what they say is they want returns. And if you go to a hospital and then the laboratory technician tells you that you are paying X amount, it becomes expensive. But you are not questioning the value for your money. Now, one advantage of molecular testing is the capacity to multiplex. Yeah. For instance, if you go to the developed countries, they have a centralized system. So you report with a fever, they can do a parasite site, investigation. They send sample to the virology lab for that. If they can't, they send sample to several labs to be able to do something. It's not like that in the developing world. You don't have systems for sample storage and then centralized sample query. Yeah. So what molecular tests can give you is you can have a set of multiple multiplexed array where you can query maybe all STIs or all febrile pathogens or all respiratory viruses at a go. So the patient wouldn't have to sample, pay to this lab to run this assay, then pay to another lab to run an assay, then pay to another one to run an assay, because he's not getting a proper diagnosis. Yeah. So sometimes it's also up to the policymakers to bring themselves up to the level of the local people to understand the value of diagnostics. Yeah. So you think you are paying more for just one test, but that test is multiplexed. And that is going to give you several other results, which will give you a better diagnosis. So I think that's how we need to explain to them. Yeah.
Well, that's a great answer. Finding the language that becomes the policy trigger for the policy people. Nicholas, any input into that? What language should we be speaking here to these stakeholders to engage them?
So I mean, again, not being a clinician, not being in Africa, right? I guess my mission's been through the network trying to understand the situation and actually have a very similar understanding to somebody who wrote in Maimuna, who just wrote in the chat that we'll need to have a strategic private, public and development partnership. And I think I agree with that. It's about government, it's about NGOs, and also it's about defining the niche. So it might just be that super accurate molecular diagnostics is not what you need everywhere in Africa.
Yeah.
So you start with clinics, you start with particular, applications, and that's where I think we'll start the implementation. So it's about finding the right case scenario and finding the right funders, probably public, but then perhaps private stakeholders in there as well. I completely agree with that comment.
Yeah. I mean, I think one of the slides you were showing yourself and Francis was of a blended kind of model where these would be used in tandem with existing technologies, and perhaps building that socioeconomic case, framing it in the socioeconomics, what they would save across their healthcare system, how it would benefit them in that ways might be a way forward as well. And Dr. Diop is from the African Development Bank, so I'm not surprised to hear that comment about this, the pull for the private, public and development partnerships. And we're totally with you on that. Dr. Julie Barlin from the University of Sheffield, and thank you, Julie, for the tweet the other day about our almost 10 years of the ISNTD. We really appreciate that. We're penning a response to you actually as well as we speak. But Dr. Barlin from the University of Sheffield has excellent questions and responses. I agree very much with finding the need and prioritizing according to local issues and needs. So just open it, and that's in very much in resonance in terms of both Francis and Nicholas's comments. In terms of the local kind of area of that, we've talked about the demand side there somewhat in terms of the entrepreneurs, the investors that are needed to kind of drive this forward, make it sustainable. It's not going to happen overnight. That's the kind of demand side when it comes to investment in entrepreneurial activity. On the supply side, the African Development Bank figures show that there are nearly 650 tech hubs across Africa, and I'm talking, their definitions are accelerators, incubators, university-linked startup support labs, maker parks, in some cases co-working sites. Out of those 650 tech hubs, Egypt, Nigeria, Kenya, and South Africa, those 4 countries account for more than a third of those, but then most countries and regions across have tech hubs in one form or another. So the embryonics are certainly there in terms of the local intellectual capacity, as it were. Young people tapping into that is going to be one of the issues, and that's what I kind of wanted to ask and weave in one of the questions that we've had. One of the questions that we had was, I believe, from Dr. Stephen Bramia in Canada, and he was asking what are the plans in place to train scientists or those communities in rural areas of developing nations to take advantage of these highly advanced diagnostic technologies. The kind of training gaps. Are you, as the Digital Diagnostics Network, involved in reaching out? You mentioned the multi-sectorial kind of nature of DIDA. Are there plans to outreach, plug any training gaps, increase capacity in that way? I'll ask Nicholas first because he's here in Imperial.
Yeah, no, it's a very good question. To be honest, I think those are things that we discuss within the network, and training is indeed important to be implemented, particularly when looking at new technology, because there's definitely going to be resistance to new technology, and that's how new diagnostic tests fail. We're very aware of that. So, the idea is to try to get as much as possible the demand from the actual field, not just design what we think is right. And that's why we've got the network. But I don't think I can reply too much more than that not being on the field.
Francis, you were there in Ghana. That's a great answer, Nicholas. We appreciate that.
Well, what I'm going to say, it's unpublished and it's basically anecdotal. Yeah, so I think the entire curriculum of laboratory technicians probably needs to be updated. Yeah, you find lots of deficiencies in, for instance, like modern trends of diagnostics tends to be lacking in their curriculum. So they're trained, they come out when they go to the hospitals, and they really cannot use or keep up with some of these sophisticated equipment. For instance, molecular testing. You get dozens of them who don't even know the basic principle of PCR. So let alone analyze data. So that's why you have lots of, there are lots of grants. We get lots of donor funding. We get lots of people sending equipment to hospitals, but they're just lying there. Because one, the people who are supposed to use them do not know how to use them. Yeah, they don't know how to use them, and they're just lying there. Yeah, so I think going forward, policymakers should try and then, give them refresher courses or update them or let them take, courses that would help them understand all these so they can use these things. And probably when they start using them, then they would probably see the need to advocate for more, and we could see a transformation. Yeah.
Okay, so there's a responsibility for the educators to
retrain,
retrain, and address this gap.
Yes, exactly.
Appearing. That's a fantastic answer, actually. Nicholas, would you like to add anything to that in terms of from the Imperial perspective?
No, I completely agree. I think very well said, Francis.
Yeah, I thought it was very well said as well. Brilliant. In terms of that's the kind of funding kind of thing out of the way, I guess, for now. The limited time, so it's kind of the funding and the kind of local voice and the local training and the asset that is the young people there right across Africa, connected, socially mobile, and how they fit into all of this new economy, as it were, as it's growing. Just turning to regulatory harmonization, or the kind of regulatory landscape, as it were. We've seen, I think, a couple of days ago, when people talk about digital, there's this kind of automatic pull in the mind towards AI, issues on artificial intelligence, probably because it's been given more prominence in the media, characters like Elon Musk, the whole kind of driverless cars thing, the whole artificial intelligence. It's really captured the mind of people when it comes to this digital space. And then people kind of shy away, or they're not that aware of this side of the digital spectrum in terms of this kind of the diagnostics that we've been talking about. But what I wanted, there's a kind of parallel there in between the two, in the sense that when you look at AI, only a couple of days ago, UNESCO have on the 21st of June, they put forward a consensus-based charter, which is now going to go to a country vote for all the member states that are part of within UNESCO's framework. It was intergovernmental meeting, and it was relating to the recommendations of a new charter on the ethics of artificial intelligence. So you can, from there, you kind of infer that there's a lot more of a kind of almost a real kind of momentum and drive when it comes to AI, to the point where we're discussing ethics at country intergovernmental level and coming up with a definition of that. So what I wanted to ask is, how do you feel about the regulatory kind of landscape that digital diagnostics is in at the moment? Is there harmonization across regions? Africa as an example. Is there work to do on that? What's your input into that? What's your kind of take on that? I'll ask Francis first.
Well, give it a little bit of thought. Well, it's true. So when you mention digital, everybody will think, oh, it's, it has to be sophisticated, it has to be, high tech and stuff. But it could simply even mean that not queuing to pay your your bills when you visit the laboratory. Yeah, one of the things that deters people, especially, and I had a personal encounter when I was on outreach to one of the villages, was that people want to go and do business. People want to go and sell. They don't see why they should queue in the hospital for several hours just to pay for a service that they would pay for. So, and you have in Kenya, for instance, they have all these mobile wallets that you can pay your hospital bills with and not having to queue at all. Yeah. So what if we could incorporate these small platforms just to pay bills, or you wouldn't have to queue, just go and pay and that's it. Or you come to the hospital, run the test. You don't need to wait for the result. I would send you the results in the house. And get policymakers to pass this and regulate. These are not things that are difficult to regulate. These are very easy to regulate. So these are small things or small steps that we could take just to make the life of patients or people seeking healthcare a bit more accessible. Even booking for a specialist before you go to the hospital. Sometimes you can go and wait for the clinician for hours before he even arrives. And the queue, you could wait for up to 6 hours, even in the cities, to see a clinician. Yeah, we could have this simple like book by appointment basis where you just book and you go to the hospital. And so these are small things that we could use digital diagnostics for that would actually save us the trouble of having to go around in circles.
Yeah. So I get what you're saying. There's these smaller steps at a societal level.
Exactly.
I mean, moving out of that's a great answer, and I thank you for that. Moving out of that into kind of a regulatory landscape when it comes to the development of a new product, as it were. You both mentioned assured, the WHO guideline. Do we need to modify that and add some more letters to that to bring it into the digital age? Do you think it's fit for purpose? And that's quite a big question to ask you, but you're on the spot. I'll ask Nicholas this first. Is assured fit for purpose for the digital age?
Well, no. And actually, the first thing I said after saying the letters of assured, I think, was added connectivity to that, which, I mean, you're absolutely right. So because you talked about regulation, there's many different areas here. There's the actual test. Does it reach the sensitivity specificity that you need? And there's the whole connected area where you need GDPR, you need to be secure. And I guess we're kind of facing that because I didn't mention, but at the moment we're going through a commercial enterprise look at implementation here in the UK at first. So, step by step, but we're going through all the process of CE marking, of MHRA, and, it is quite, of course, heavy. And looking at other countries, definitely it's something that needs to be taken on a case by case. And the data part is, of course, something that raises eyebrows if you're going to be collecting data everywhere. So it needs to integrate with whatever system is in place there. Here the NHS, over there, I guess that's to be discussed. Yeah. There's a lot of regulatory considerations in there. I'm hoping that public organizations such as those that might contribute to funding would be able to guide with that, NGOs or perhaps governmental organizations.
I mean, it's certainly reflected in the kind of wide array of different disciplines and different kind of sectors involved in this your own diagnostics network, the digital diagnostics network. So perhaps in the regulatory sphere to get different inputs wouldn't be a bad thing. In fact, this intergovernmental meeting that UNESCO have chaired, and they're the ones that are putting forward this new world charter for ethics of artificial intelligence. One of its kind of redeeming factors, as it were that they haven't just relied on G7 countries for their definition of ethics. They've really involved lots of governments to get lots of different viewpoints so that the ethics kind of definition itself is not biased in any one direction. It's kind of reflective of the world we're living in. So I think there's room for thought on that.
Aubrey, very, very quickly, Aubrey just mentioned apparently there's a movement to harmonize regulation over in Africa. Thank you, Aubrey. I wasn't aware.
There you go. Yeah, exactly. So this is something that's being looked at, and it's certainly something that needs to happen. And when it comes to perhaps digital is now forcing the issue somewhat, or at least accelerating that issue. So I mean that's great. Francis, would you have any input on that in terms of?
Well, I think the the term assured is even subject to revision. I think we are now at reassured. Yeah, reassured. And reassured means, I mean, the RE just means real-time connectivity of data. Yeah, so it's something that the entire populace is, discussing to try and put some ethical control on this, because if we are going to have real-time connectivity, there is going to be the data transfer, and this is being regulated. Yeah.
Exactly, and the entire, all the ethics nuances that come with it, everything. Absolutely agree. So it's a vital part of this whole reimagining process that we're putting ourselves through. You mentioned Aubrey's questions there. So he'd asked some questions yesterday, actually, that he'd sent through, and I'm going to ask those now. So one of the questions is, when might we expect the first point-of-care digital diagnostics to become commercially available, and how much will that cost? What will be the cost of that?
That's a question for Nick, actually.
I think that is a question for Nick, because he's,
Well, it depends what's meant by malaria point of care. I mean, if we're talking about rapid tests, that's a different thing. If they just lateral flow tests, but I think the real challenge here is to have molecular point of care. In which case, I think we're still a few years down the line, because we want to do this right. We want to make sure we talk to early adopters, want to make sure we design for Africa and not just design in the lab and force implementations. That's not how it's going to work. We've been taught that in the past. And so several years is as far as I'm going to go in terms of guessing, because there are many levels to get there.
And any idea on the costs? Are we allowed to ask you that?
So I think it's foolish to think that we're going to get down to the cost of lateral flow tests, because let's face it, lateral flow is a piece of paper. Whatever you do for molecular detection will be more than that. Here we're talking of chips that, of course, can be significantly cheaper than your large instruments, but of course it's not going to go down to a piece of paper. Therefore, I think we're looking at something around the range of $5–10. And that's why, I guess that's the actual challenge here, is that it's kind of a new niche, isn't it? You've got the very cheap lateral flow test, you've got the expensive test that needs a lab. We're trying to identify a new market in the middle, which of course will have its own case scenarios, as I said earlier. So that's where I think the cost needs to lie, somewhere in the middle. But because it's Africa, it needs to be, of course, affordable.
Yeah, absolutely, and be sustainable on that basis. Dr. Diop from the African Development Bank, in reference to this whole regulatory harmonization framework, she's made a point that in Africa, we will need also to think about a strong regulatory framework, as we still have issues on data protection. And just coming back into that ethics thing on data for a second, Francesca Pfeiffer from Imperial College, I very much agree about what was said about policymakers and driving the demand. A focus is needed on implementation partners, on influencers, and on building health knowledge about the benefit of digital diagnostics. What you can do with data is a great starting point, and I agree with Francis. So there's a tick for you there, Francis, in terms of some of the points that you'd made earlier. So I suppose that's kind of, the kind of partnerships that need to happen, is raising awareness, putting it into a language that not just policymakers, but the youth can understand, or that demographic that's going to be there involved in those digital activities, that they can actually understand as well. There was a question again from Aubrey. In terms of incentivization of that particular section, what sort of evidence might policymakers and funders need to make this paradigm shift? Because it is a big shift. We are talking about reimagining the entire quadrant-based healthcare model, not just the diagnostics quadrant, but this has implications for the biotech sector, for the pharmaceutical sector, for the generic sector, through supply chain synergies in terms of digitalization, all sorts of impacts. So we are talking about reimagining this healthcare model that we're all kind of stuck in. What sort of evidence, what sort of will policymakers, funders need to make this happen? We have slightly answered that before, but I mean, any other further input on that from either of you?
Okay, so I believe one of the challenges that faced by the lack of diagnosis is that if you're looking for some data on certain diseases, you won't find the data. You either get very old data, or they just try to assume from the burden, oh, it's just about this based on the local report. But the population-wide study has not been done because there are no diagnostics for these. So with digital diagnostics, where these things are available, and you get patients to use these on a regular basis, I mean, data could be put on a hub, which could be accessed by policymakers just to know where they should target control or where they should need to focus next or stuff. Yeah. So I believe digital diagnostics hold lots of prospects in this area. That's why it's really important.
I suppose you're alluding to the real integrated national disease surveillance program,
exactly,
making it absolutely cohesive in all the different parts.
Exactly.
So there's another framework for digital to kind of get involved in. Later on in the year, in October, out of interest, we'll be running our vector control and disease surveillance meeting, ISNTD Bites, which is our annual conference. Sorry for the pun, but it's about vector control and disease. So one of the panels we've got set up there is looking at exactly that recommendations towards anyone looking to build a national integrated disease surveillance kind of program. What are the technologies and what are the areas that they should be looking to incorporate in? So I'm sure we'll be in touch through Aubrey and Francesca and yourselves for that, but that's definitely one of the biggest sessions we're going to be running there. You know, if you had to give a report to a government, this is what we would recommend from the consensus. But this is going to be definitely one of the points we're going to be putting into that session. So there's another position for the digital revolution to take hold on, which is fantastic, really. We've been getting a lot of kind of feedback from people. We're joined by Dr. Ikram Guizani from the African Leishmaniasis Consortium from the Institute Pasteur in Tunisia. So, you know, I know we've talked about malaria here in a disease setting. You mentioned dengue and some of the other kind of areas of application. But are you both open to different disease kind of focus groups coming to you to talk to you, to liaise with you? Are you both open to that?
Yeah, sure.
Good, because we've got, you know, we've had Derek Robinson from the University of Bordeaux. France is asking if we can get in touch with you over the next few months.
Yeah, sure.
That's it. So I'm sure you'll have something to discuss there.
But perhaps I can quickly add, you know, the way you can see the technology is electrochemical imaging. So, you know, I talked about DNA. There's a lot of things that can be done with amplifying DNA on any kind of targets, right? But there are other biomarkers that we can look at, and we're very happy to talk about new areas of research.
That's very, very good and very good to know. And I hope from our audience perspective that there is some traction with that and some follow-up, because it's a very open group. We've really enjoyed working with both Aubrey and Francesca and in various settings. So that's really good. That's brilliant. So that's very good to know. We're coming up to the end and of the allocated time, and I'm under a bit of time pressure from the powers that be on our side to make sure we end on time. If you could see what I could see. So I'd like to say thank you very, very much to both of our speakers today. To Dr. Francis Krampa from the University of Ghana. We salute you. Thank you very, very much for such a brilliant presentation and the applications and the answers that you gave in the Q&A. To Dr. Nicholas Moser from here Imperial College in London. Absolutely blown away by the meeting of two worlds, as it were, as you put it yourself, the biologists and the electronic engineering world. Godzilla versus Technico, that type of mashup. Fantastic. Look at the results. Francesca's put the website up there for you. I'd like to say thank you to Francesca and to Aubrey as well. You're getting a lot of thank you from Dr. Guizani, from Dr. Farisi May, who's saying thank you for your time, Dr. Francis and Dr. Nick, extremely informative. They've absolutely, I think the audience has found this very interesting indeed. So again, thank you. I think that's gonna bring it to the end for us here today. There's never enough time. There's so much you could have a session just on regulatory harmonization. on the technical kind of framework, just on the kind of funding, the policy frameworks that are out there. And I'm sure this is the first in a series of sessions that we're planning out with the Digital Diagnostics for Africa Network. So please do watch this space, everybody. This really, from our opinion, is one of the hottest spaces to watch for global health, without doubt. And it's all part of the reimagining a future post-COVID, as it were, and the reality of that. You know, what is it going to take to achieve healthcare equity? These are huge questions. What's it going to take to achieve inclusive prosperity as economies bounce back? This is one of those moments in time where we're either going to get it right and this is going to work out, or we're not. So these players here, these actors here that are instrumental in this, and certainly we cannot underestimate the importance of the Digital Diagnostics Network for Africa. We salute you. We're delighted, honored to have you on this session. Thank you very, very much. Huge shout out to the audience. Thanks a lot, guys. Take care. Stay safe, everybody, wherever you are in the world.
Yeah,
right?
sure.
Thanks a lot.
Thank you, everyone. Thank you.
Bye-bye.
Bye-bye.